All right. Hello, everyone. Welcome and thank you for joining us today. It is now 1 o'clock, so we will get started with today's webinar. I'm Megan Weber, an extension educator here at the University of Minnesota, focusing on aquatic invasive species. I will be your moderator today, and I'm joined by Pat Mulcahy, our program coordinator, who is helping everything run smoothly in the background, and our speaker, Mike Szymanski. I will give a couple of quick housekeeping items, and then I will turn it over to Mike for his presentation. So we are recording, and this will be available on our YouTube channel. About a week after today's event closes. So if anything comes up, you run into technical issues, aren't able to stay for the whole thing, don't worry, you'll be able to catch everything later. We are using Slido for Q&A. There is a website showing on my screen now. It's slido.com, and there's a code GENE21. And you can enter that to join the Q&A. And this will give you the opportunity to enter questions at any point during Mike's talk as they come up. And you can also upvote other people's questions that are particularly interesting to you. And that way, in our limited time for questions, we're able to get to as many as possible that are of the most interest to the most people. A very democratic way of deciding which questions will get answered live. And let's see, you will all be muted for the duration of the webinar, but feel free to use that Q&A. If you are having troubles accessing Slido, go ahead and put your question in chat, and Pat is here to help. He will get those transitioned over to Slido so that they become part of our Q&A pool that way. I think that is all that I have for our housekeeping. So with that, I will turn it over to Mike to introduce himself and begin his presentation. Thanks, Mike.
Great, thank you, Megan, and thank you for the invitation to present, and thank everyone in the audience for being here to tune in. Again, my name is Mike Szymanski. I'm a professor at the University of Minnesota in the Department of Biochemistry and the Biotechnology Institute. I've been told we have a very diverse audience, and I think that's great. I'm going to keep this presentation fairly high level. It ought to take somewhere between 30 and 40 minutes, so we'll have plenty of time for Q&A at the end. To start out with, though, I wanted to make sure everyone is on the same page at a really high level. And the meat of this talk is going to be about genetic engineering. So what is genetic engineering? To sum it up, I'd say genetic engineering is the rational alteration of an organism's genetic blueprints. That could be transgenesis. You may have heard about transgenesis, which means you're moving a gene from one type of organism to a different type of organism, but it doesn't have to be transgenesis. It could be deleting a gene from an organism. It could be going in and just rewriting the DNA sequence at one or a few genes in an organism, or it could just be taking genes in an organism and moving them to different places in the genome. You can think of the genome like a book that normally reads start to finish. And if you take a paragraph or a chapter out of a book and move it somewhere else, it can change the meaning or the impact of that book. Similarly, as we rationally move genes around in a genome, it can change the behavior or the form and function of the organism. So one thing that genetic engineering is not is it's not scary. It's not scary to me, and I don't think it should be scary to you. It's a type of technology that has a lot of applications. If there's one silver lining to the COVID pandemic that we've been living under for the past year, maybe two silver linings. The first one is it's demonstrated the huge upside of biotechnology. When you look at the breakneck speed and effectiveness that the modern mRNA vaccines were brought to market. It also has increased the base level understanding of molecular biology and virology. Almost everybody now has heard of mRNA, which as a molecular biologist, I think is great. The cartoon I have across the middle of this slide shows something that we call the central dogma of biology. It describes how information is converted from DNA, which are like the long-term lasting blueprints of life, to protein, which are the functional workhorses in a cell. And the arrows that connect the two, DNA to RNA to protein, do not designate a transmutation. DNA does not become mRNA. mRNA doesn't become protein, but each one encodes or directs the synthesis of the next in the chain. And so a really good analogy for this is a printing press. DNA would be the pages of the printing press, the metal letters that are arranged In genetic engineering, we could move those letters around to change what's stored long-term in that printing press page. DNA is heritable, it's stable, it's going to be inherited by the progeny when organisms reproduce. And organisms have thousands of genes. You can think of each gene being a different page in a printing press. Now, while all the cells in an organism have the same genes, not all of the genes are turned on at the same time. And when we say a gene is turned on, we mean that that DNA is being converted into RNA, or it's being templated to produce messenger RNA. So in this analogy, messenger RNA is the paper newsprint. It has the same information as the metal printing press page, but it's in a more transient form. We know that the paper is going to last for a day or two and then it'll break down. Similarly, with the messenger RNA, it has a short half-life. It stays around in a cell for a few hours or a few days before it's degraded by the cell. And different cells will be turning on a different subset of their genes at any given time. From messenger RNA to protein, again, you want to think of if those newsprints in the middle of the slide are encoding blueprints for a house, you could say the paper is directing you how to build the house, but it doesn't mean the house is built out of newspaper, right? It's just the instructions. And in the same way, protein, the instructions to make a particular protein is encoded in a particular mRNA, but it's not, the proteins aren't made out of mRNA, or the mRNA isn't made into proteins. And so, I'm going to be talking later in this presentation about turning genes on and off. I'm going to be talking a lot about genetic engineering. The genetic engineering that I'm referring to is changing the DNA. So it's a stable and heritable change to the blueprints of an organism. But again, biotechnology, genetic engineering is not scary. I've received an mRNA vaccine. Probably half of you or more have received an mRNA vaccine. I was kind of proud of that for a few days afterwards, I was saying, I'm not 100% human right now. I'm partially transgenic. And I'm not trying to argue, there's some conspiracy theories that the mRNA vaccine is going to make you a GMO organism. It's not going to alter your DNA. But for a while, your cells were delivered the message to enable them to make a protein that's not encoded in your genome. I think that's really cool. The protein that was produced is one of these coat proteins of the coronavirus, and that enabled your body to mount an immune response against that protein. So biotechnology is powerful. It has a huge upside. Now, the main talking points for this webinar, I talked about what is genetic engineering, but we're going to go into a little bit more detail of what is genetic biocontrol. How can we use genetic engineering to combat invasive species? Does it work? Is it wanted? And how long will it take before it's a realistic solution? So first of all, what is genetic biocontrol? In a nutshell, genetic biocontrol is using genetic engineering to turn a pest organism into the pesticide. Typically what we'll do is we'll start with, in this example on the left, I'm showing carp. You'll start with carp. You'll make a genetic biocontrol agent, which is a genetic engineered version of the carp, and you'll re-release that back to the wild. Yes, you're re-releasing a version of the invasive organism back into the wild in the places where it's already invaded. But that organism, by mating with the wild versions of that pest, will actually drive down population numbers. Genetic biocontrol is not a silver bullet. I imagine that when it is applied, it will be applied in combination with other methods as an integrated pest management plan. So in the slide here, I'm describing, physical methods could be used to net or trap an invasive species to reduce its numbers, and then release of a genetic biocontrol agent can help eradicate it locally. There's a lot of potential advantages for genetic biocontrol. It's chemical free. I'll show you some public opinion slides in the end that says if there's one thing that the public doesn't like more than biocontrol, it's chemical control. Chemicals tend to act non-specifically. And so chemicals that, for example, would be effective against carp would kill a lot of other native fish in that body of water. Because the released genetic biocontrol agent is gonna work to suppress the population by mating with those wild organisms that are out there, it tends to be very species specific. A released genetically engineered carp will try to mate with wild carp, but it's not going to try to mate with native fish like walleyes or northern pike, so it's not going to impact those fish directly. We know from experiments that have been done in labs and the few that have been done in field trials that these have the potential to be effective solutions. And we know that they're scalable as well. engineering biology is neat because it's the one engineering medium that once you make the machine, it makes more of itself. Many of the genetic biocontrol agents will be stable and have heritable mutations. So taking your first engineered agent and producing 1,000 of them is as easy as fish husbandry or insect husbandry would be for that particular organism. There are several different types of genetic biocontrol. The first and what's considered the gold standard by which everything else is compared now is called sterile male release. Now, this has been around for several decades and it's been quite effective. I argue that it's the most effective pest control method ever deployed. The way it was deployed in the past doesn't use genetic engineering per se because it doesn't use rational changes to the genotype, but instead it uses these large irradiation chambers where larvae or pupa of an insect are put into these chambers and they're bombarded with radiation to sterilize the males. And then they're released by the millions out in nature, and those sterile males will try to mate with wild females who then won't have any offspring. And in that way, it can crash a population. Now, I argue that this is the most effective type of pest control ever deployed because it's actually eradicated a pest from an entire continent. The map on the left shows the old range and the current range of the New World screwworm, which was eradicated from North America using a campaign of sterile insect technique. Now, the reason this isn't used widely for all invasive species is that it doesn't work well for some species. Oftentimes, the dose of radiation that you need to sterilize an organism is so close to the dose needed to kill the organism that by the time you deliver enough radiation to sterilize the organism, they're so sickly that they can't compete with the wild males if they were released to the environment. And so excitingly, in the past few years, there are several new approaches that are enabled by genetic engineering that would work the same way as sterile male release traditionally has, but they are more robust and applicable to a diverse array of species. Another type of genetic biocontrol is called sex ratio biasing. Most populations have close to a 50-50 mix of males and females, and that's required to maintain the populations at the numbers that they exist at. It's possible through a number of specific methods to create genetic biocontrol agents, such as the male represented with this green circle in the bottom left of this slide, that are unable to have, for example, female offspring. And so with the release of these genetic biocontrol agents, you can, and with iterative releases, you can eventually skew the ratio of males to females so much so that there's not sufficient females left to support the population will crash. Again, several different ways, specific ways to implement sex ratio biasing. You may have heard of daughterless carp, which was the brainchild of Ron Thresher, pictured on this slide. But there are other ways such as female lethality, or even manipulating sex chromosomes to create Trojan males or YY chromosome males. A third type of genetic biocontrol, which has received a lot of news in the popular press and in the scientific literature in the past 5 years, is something called gene drives. Now, if you look at the inheritance tree on the left, that's an example of normal inheritance, where if there's a parent that has a gene that creates this green color, and a wild type, let's say, doesn't have that gene, so they're gray. Well, half of the offspring are going to inherit that gene from the one parent, and half of them won't. And in that next generation, again, half of their offspring will inherit the gene, half of them won't. And so you have this 50% inheritance each generation. And that keeps the frequency of that gene in the entire population more or less constant. Gene drives, however, which are can be, they've been around for several decades in concept, but what's changed in the past 10 years is that they're much easier to engineer using modern tools of precision genetic engineering. These amount to a selfish gene. We can design and integrate into the genome a selfish gene, and that selfish gene, if it's inherited in offspring and it's inherited at a 50% rate, it'll actually find the corresponding gene that was inherited by the other parent, and it'll cut it out and do a copy paste. So it'll replace itself in that gene from the other parent. And now that progeny all have 2 copies. And so 100% of their progeny will have the copy, and 100% of their progeny will have the copy. And so you're getting an inheritance rate much greater than 50%. And that can allow that selfish gene to spread like wildfire through a population. Now, gene drives can be used just to push an interesting trait through a population, Or you could even spread a trait that's bad for the population. And so there'll be an initial burst where that deleterious gene is spread through a population like wildfire through mating. But then once a large enough proportion of organisms in that population all have that gene and they try to mate with each other, all of a sudden none of the matings are going to be successful and the population will crash. And so, these are kind of painting types of genetic biocontrol in broad strokes. There's a dozen or more specific types of genetic biocontrol that, they'll fit loosely into one of those three groups, but they're important in key ways. So one important criterion to consider when you're evaluating a specific genetic biocontrol approach is how persistent or invasive is it going to be. Some controls approaches like SIT stands for sterile insect technique. That's that male sterile release that I mentioned. These are not persistent at all. If you release a bunch of sterile males, they'll mate unsuccessfully with wild females and crash the population that way, but they have no way to reproduce themselves. So they're not going to make more sterile males once they've been released. Oftentimes these genetic biocontrol approaches at the left end of the spectrum need to be released in much greater numbers and often iterative releases. So maybe 1,000 genetic control agents every other week for a few months because they're not going to hang out. On the other hand, organisms like gene drive control agents, releasing even a small number of those will allow the genetic modification to spread through the population. So they don't need to be repeatedly released, and instead they'll be self-sustaining. And so that's an important parameter to look at when you're evaluating a specific genetic control. How persistent or how invasive is it? But there are other parameters that are important as well. Just like you may have heard about resistance to antibiotics, genetic resistance to genetic biocontrol can evolve in wild populations. And every different type of genetic biocontrol may have a different route that resistance can evolve. So understanding those routes by trying to break, I'm an engineer. So when we get something to work in the lab, the next thing we do is we try to break it as many ways as possible so we can build in safeguards and prevent those failure modes. But understanding what are the possible failure modes, what are the risks and consequences if resistance does evolve. And the last bullet point I have here is what specific aspects of the biology of the control agent are relevant. This is a cutting edge area of research, but we know that biology is incredible for its diversity of behavior, form, and function. You may have a genetic biocontrol approach that works great in insects where the females only mate once and never again for the rest of their life. A sterile insect technique might be very effective in that case. But if the females mate oftentimes with many different males, the sterile insect technique would be less effective because they might mate with a sterile male and then tomorrow go mate with a wild type male and go ahead and have offspring still. So the biology of the target organism is an important thing to consider as well. So does it work? I'm trying to illustrate here, and this is not the most comprehensive representation, but in these three broad stroke categories of types of genetic biocontrol, advances are being made in insects and aquatic invasive species and mammals as well. It's fairly straightforward to design a genetic biocontrol for any organism now. Several have been published as proof of concepts in laboratory experiments where they're in a contained laboratory, they're not getting out to the wild, but we can show that in concept the strategy works in controlled matings. I'll give you an example of that in a few slides. Several genetic biocontrols have been field tested in insects and in fish as well. And I've made this in-use one for insects, kind of a duller red. That's, I'm referring to the sterile insect technique, which is widespread. It's used all over the world. I dulled it a little bit because it doesn't use genetic engineering. It uses radiation to sterilize the males. But again, it's been used in commercial settings wildly successfully. So the next 5 slides are going to be a little bit of I'm going to nerd out a little bit for those of you in the audience that are more technically minded. I'm going to tell you the details of a particular type of genetic biocontrol that we call engineered genetic incompatibility. This was developed by students and postdocs in my lab. And what it amounts to is engineering a speciation event. So we take a wild type organism. Here I'm representing a carp again, but I'll show you some insect data in the coming slides. And through some pretty straightforward genetic engineering, we create the biocontrol agent. Here I'm representing it as a blue fish. It's not a blue fish, so it looks and acts and behaves exactly like the wild type fish would. What's different about it though, is that it's incompatible. It can no longer crossbreed with the wild fish. So you might think that sounds a lot like sterile male release, but it's not quite sterile either. Males and females of the engineered type can mate with each other and have viable offspring. And we're excited about that because it makes it much more scalable. Once we engineer what we call an EGI organism, we can just propagate them in the lab using normal husbandry techniques to make as many as we would like. But eventually it could be used in a way that's very analogous to sterile male release. If we separate the males from the females and only release males to a body of water, they would be incompatible with any female that they mate with. Basically, their sperm is acting like a pesticide, and any egg that they fertilize with their sperm will not develop to adulthood. So we've published recently a proof of concept of this in flies, and so I'm going to change gears a little bit from aquatic invasive species to this data with flies, but in concept it works the same. The way EGI works is that we're perturbing normal patterns of gene expression, or which genes are turned on. We do this using a new set of molecular tools called programmable transcription activators. Transcription is a science jargon for the process of turning a gene on. And we can use derivatives of the CRISPR-Cas. You may have heard of CRISPR-Cas. It's the hottest new tool for genome engineering. We don't actually use it to engineer the genome in this case. We've hampered it a bit by making it unable to cut the DNA. But what it can do is it can go to any gene that we direct it to and turn that gene on, start making those news copy prints from that template in the DNA and make thousands of copies of the mRNA for a particular gene. Now we've engineered the EGI strain so that that aberrant pattern of which genes are turned on doesn't happen in this strain. It only happens when that strain interbreeds with the wild variety. It turns out all of our genes are turned on in specific cell types at specific times. And if you have a gene that should only be turned on in a developing heart cell, but you turn it on to really high levels in a developing brain cell, that's going to be bad for the developing organism. And so that's in a nutshell how this technique works. We're not introducing any genetic toxins. There's no poisonous genetic constructs. All we're doing is kind of mucking up the regulation that normally controls which genes are turned on in which cells, and that leads to hybrid lethality. So it took us approximately a year of work to put together all the genetic components and build an EGI line of flies. Here I'm representing it by this yellowish green color. And the way that we tested it first was by doing a series of controlled matings where we would combine males and females of different types in tubes, remove the adults and let them go for about 2 weeks and see what progeny emerged. I'm going to show you an example experiment on this slide. So this is a time course video from a 15-day experiment. The genotypes of the male and female parents are illustrated below the video. You see on the sides we have the same type mating, EGI with EGI or wild type with wild type. And when all those flies disappeared, they didn't just die. What happened is we removed them from the vials because what we're really interested in is what happens in the next generation. In those first 3 days, the males and females were allowed to mate, copulate, lay their eggs, and we're watching what happens as those eggs develop. So the first thing that you'll notice is if you pay close attention to the tubes on the side, you see a lot of motion. That's the larva swimming around. Insects go from a larval stage, several larval stages, to a pupal stage before they emerge as adults. And you see a lot of activity when the male and female parents were of the same type. Now, if you look closely at the middle tubes, you will see some movement still. Some of the larvae are viable here. And the important thing with our technology, we have the ability to tune when the hybrids die. Sometimes they'll die at the larval stage. Sometimes they'll die at the pupal stage. Sometimes they'll emerge as adults, but they'll die before they can mate or they'll be infertile. And that turns out to be a really important parameter for this technology, because for some target organisms, it's better to kill the hybrids really early. Sometimes it's better to wait until pupal stages or later so that the hybrid larva can outcompete for resources with the wild type larva in the same environment. And so what you're going to see in a bit here is this is the end of the experiment. We can count how many adults emerge. And in this case, you saw no adults emerge from either of the hybrid crosses in the middle, but we had plenty of adults in both of the side vials. And we can count these adults and represent the data in a dot plot graph as shown on the left here, where we're showing the x-axis, the bottom axis shows the genotype, either wild type or engineered genetic incompatible of the male and female parent. And then for different replicate experiments, we count how many adults were produced. And so this is an example of 100% hybrid lethality. We got no surviving adults from either of the hybrid crosses. Now, the right side of this slide shows something that we're really excited about. This is representing the data in a little bit different way, where we're using a heat map. So the color of a square, if you draw a line between a name on the left axis and a name on the top axis, the color of the square corresponds to a number on this color bar that describes what the average number of offspring were that survived to adults. So Oregon R, that's the wild type. That's the wild version of fruit flies that we use in the lab. And you can see that when an Oregon R male and female mated together, they had roughly 100 offspring. These other three names are three different EGI strains that have a different genetic design. And importantly, when we mate the male and female of the EGI of the same design together, we get anywhere between approximately 100 and 40 surviving adult offspring, or offspring that make it to adulthood. But the beige color in this grid is actual experimental data that we collected as well, but the average number of offspring was all 0. And what that means is that we can engineer multiple different EGI lines that are all 100% incompatible with wild type, and they're 100% incompatible with each other as well. That has huge ramifications for dealing with genetic resistance. You can imagine a strategy where if you have just 2 EGI lines, so on the previous slide I'll show you, we already have 3. Actually, we have about 16. But even if you just have 2 EGI lines that are mutually incompatible, you can have an iterative release strategy where if you have a wild population, you release strain 1, the males only, they're gonna suppress the population. And then you can release strain 2, and then strain 1, and then strain 2, and then strain 1. And the reason this is important is that let's say there is a low level of genetic resistance that evolves. Some of these wild type will have diverse genotypes. And if one of them has a genotype that makes it so it's not 100% hybrid in viability, but that some of the offspring between that rare mosquito and this green mosquito survive, and I'm representing them here as a pink one, then that would be an example of genetic resistance. But since that resistant offspring just inherited half of its genetic information from a strain that we know is incompatible with the next strain that we could release, we're basically sneaking in a susceptible gene that's going to guarantee these pink flies will not be resistant to the next release. And if you keep alternating between 2 releases, this is something that's considered in the jargon of the field, negatively correlated cross resistance. It was kind of a golden fleece in the field of pesticide research back in the 1980s, but people largely gave up on it because it was very difficult to achieve. It refers to any instance where you have 2 pesticides and gaining resistance to one pesticide automatically makes you susceptible to the other one, and vice versa. We think that we could achieve this to some degree using EGI as a control strategy. So that's how it works. What does the public think? I'm going to walk you through kind of how the perception has changed over the decades. This fundamental paper in risk analysis from the mid-1980s surveyed a bunch of people across the country and tried to describe how different technologies were perceived in terms of unknown risk and risk of dreadful consequences. And DNA technology was an outlier in both degrees. It was kind of the furthest in the upper right quadrant of this chart as being having both unknown risks and potentially dreadful risks when the public were engaged. That changed a lot over the next 15 years. You can see by the early 2000s, when people were more comfortable with biotechnology, and Ron Thresher published this paper. This was a survey around his daughterless carp technology. They looked at a lot of stakeholders in aquatic invasive species control and ranked the appropriateness of different approaches for targeting aquatic invasive species. Genetic biocontrol made it to the list, and it made it above this line of things that were deemed acceptable options, but it wasn't very high on the list at that point. In fact, the highest thing on the list was to do nothing and hope the problem goes away on its own. And what we've seen in the almost 20 years since then is that a lot of these comfort levels, a lot of these approaches have changed their ranking, in part as things that are high up on the list have been tested and realized they don't work, and also as we learn more and understand more the approaches that are further down on the list. In a survey conducted by the Pew Research Group 3 years ago that looked at appropriate uses of genetically engineered animals in society, the technology that showed the most support, with over 70% of survey takers responding that it was an appropriate use, was using genetic biocontrol to reduce populations of mosquitoes that can spread disease. So this is a slightly, there may be more support for that application than in invasive species control, but it's at least an indication that public perception is shifting, and people see this as a viable and acceptable tool to have in your arsenal of management strategies. We've done some surveying ourselves, looking specifically at genetic biocontrol of invasive carp, mostly of Minnesotans. This was a survey conducted over 2018, 2019 with over 1,300 respondents. Many of you on this call may have been respondents. We conducted it both through email at the MASERC listserv and also at the Minnesota State Fair. And we had a number of questions. There's approximately 50 questions. I'm only going to give a high-level summary here. On the bar graph at right, we're ranking the respondents' comfort level in different types of control approaches. And what we saw was that, in general, the physical methods, fishing, trapping, netting, showed a much higher level of control or of comfort than did the biological control, and chemical control options showed the least amount of comfort. Of the biological options, genetic predator introduction or pathogen introduction, genetic biocontrol had the highest levels of support. And interestingly, after just a short paragraph describing sterile release, sex ratio biasing, and gene drives, similar to what I introduced you to earlier in this talk, the level of support for those three technologies actually increased quite a bit. Some other interesting outcomes from this study is we asked which of these technologies do you think would be able to eradicate a target population? And the three at the top of the list were chemicals, release of pathogens, or genetic biocontrol. Now, chemicals and pathogens were also seen as the two most likely to harm native fish populations, whereas genetic biocontrol was the second lowest likely to harm native fish populations. Again, this is a public opinion survey, not empirical evidence. The only thing that was seen as being less likely to harm native fish populations was recreational fishing, but that actually wasn't seen to be an effective control approach at all. One neat correlation, and this is the only other data point from the survey that I'm going to highlight on today's talk, is we looked at what other survey answers would be the most likely to explain the comfort level with genetic biocontrol. And so these abbreviations at the bottom of this graph, VC is very comfortable with genetic biocontrol, MC is moderately comfortable, U is uncomfortable, VU is very uncomfortable, and I don't know is the bars on the right. And for people that answered that genetic biocontrol, that they perceive genetic biocontrol to be more effective than conventional techniques, they are overwhelmingly more likely to be very comfortable with its use. This is interesting because this effect was much stronger than the correlation between perceived risk and comfort level. And that tells us that the public are more heavily weighting their perceptions or their beliefs about the efficacy of a strategy than they are the risks of a strategy in deciding whether they'd be comfortable with its implementation. So that's what, does the public want genetic biocontrol? The survey's still out on that, the jury's still out on that. We'll see as we get closer to really bringing these techniques to the field. But how long would it take realistically until genetic biocontrol is a management tool? Well, you guys probably have heard that there are field trials going on right now with genetically modified mosquitoes in Florida. Those field trials have been going on for over a decade and a half in other countries. And so for some target species, genetic biocontrol is ready to go already. For carp, and these slides are specific to engineered genetic incompatibility, or the approach that I went into detail on, and translating that into carp. We normally have a roadmap that looks like this, where we'll use a model or easy to grow, easy to manipulate, small cage requiring model organism like zebrafish to do a lot of the testing of genetic components that we know we'll have to introduce to carp. And so while we're developing and optimizing those genetic components, identifying which genes we want to turn up too high, we'll do that in the model system. And in parallel, we'll lay the groundwork for doing that same work later in carp. So we need to establish robust carp aquaculture methods to get them to spawn year round, which is not trivial for some organisms, and to do the transgenesis year round so we can take as many shots on goal as possible. We want to develop computer simulation models and learn more about the natural population genetics so we make sure the version we engineer in the lab is actually going to work in the field. This is kind of where we are right now. We've done all of the groundwork in carp, and I'll show you some slides describing that. We've identified the lethal target genes using a model fish, and we're currently in the process of combining all those elements so that we can have a proof of concept in fish similar to the proof of concept that I showed you already for flies. Here's some of the work showing the proof of concept, in the model zebrafish, identifying which genes are appropriate for driving lethal overexpression or turning them on too strongly. Like with most genetic engineering projects, we start with a narrow set of shots on goal that we think have the potential to work, but not all of those are going to work. So we tested in this experiment 5 different genes where, in a similar way that the mRNA vaccine is introducing the message of a gene into our cells, we were introducing messenger RNA for a gene that was already in their genome into these cells, but we were introducing a lot of the messenger RNA so that it simulates that gene being turned on to a really high level. And what we found is that 2 genes in particular were highly effective at causing embryo lethality after 2 to 3 days. The top 2 images here show what a normal 1-day-old zebrafish embryo looks like. This one had no treatment at all. The one on the right was injected with just a saline solution, but it would go on to hatch and grow and be a healthy adult. The 2 pictures on the bottom show what happens when we turn on a gene called sonic hedgehog A, or SHHA, or a gene called GATA5. When we turn that on too strongly, you see strong morphological aberrations in these embryos, and these wouldn't survive to adulthood. So we've identified which genes will work in fish for turning them on too strong and lead to hybrid lethality. And we've also recently established approaches for genetically engineering carp. So we have the ability to spawn carp year-round now in the MyCirc containment lab. We can isolate sperm and egg separately, do in vitro fertilization, and then inject with a very fine capillary glass needle synthetic DNA molecules into these cells where they'll get incorporated into the genome. And we'll hatch those injected embryos and look at the fry to identify which fish have the transgene in them. In this case, we're optimizing those protocols using a transgene that encodes for a green fluorescent protein from jellyfish, just so that it's really easy to see where the transgenic fish are. And we've learned that we can make transgenic carp with fairly high efficiencies now. Now, we're not to the proof of concept yet in zebrafish. To engineer genetic incompatibility, and every genetic control approach is different. It turns out gene drives are a little bit more straightforward to engineer. EGI takes several rounds of transgenesis. And so this is our pathway forward for the zebrafish, where it's going to take 5 generations of fish before we have our stable EGI line. Now in flies, each generation takes a few weeks. In fish, it takes a few months. That's one of the reasons why we're lagging behind in zebrafish. In carp, each generation takes between 1 year and 2 years. And so already the years needed to get even to the strain that can be tested in the lab, you can tell for carp, it's a 5 to 10 year project. And after those 5 to 10 years, to have a version of carp in the lab that we can test, the next steps are to do as many tests as we can in a controlled laboratory environment, similar to what I showed you for the flies, to show that it's effective, it works the way we think it works, to do any tests that we can in the lab to predict whether it would be dangerous in the environment. And only when we're comfortable that it will be both effective and safe for environmental release would we submit a permit to a regulatory agency like the FDA, EPA, USDA, who would then need to review that permit application, look at our data, look behind the curtains, make sure everything is correct. And then if everything looks good, they will approve a field trial. With an organism like carp, which is long living, those field trials might last years by themselves. They would likely be performed in controlled ponds, outdoor ponds, where you would introduce wild type carp and then introduce the GMO carp and track the survival rate in control versus experimental ponds. And only after long and extensive field trials, where again, the technology would have to be vetted, shown to be effective and safe, would a technology be approved for use in the field by aquatic invasive species managers or water district managers. So what can we do to accelerate this development? One thing that we've done recently is we've just launched a new international research consortium that we call GCARP, Genetic Control for Aquatic and Reef Pests. All we are right now is a quickly thrown together website, but we have a small group of like-minded researchers that will meet together regularly to discuss how we can progress these technologies through increasing market-ready levels. Even in the past few weeks, I've gotten more interest from people to join this group and participate in these discussions as we try to be the rising tide that lifts all boats of genetic biocontrol for aquatic invasive species. We're also working with some new collaborators on campus at the University of Minnesota. For example, John Biscoff and his group have a strategy that we're going to test in carp to accelerate the generation time. Is there a way we can take carp that would normally require 12 to 18 months to reach sexual maturity and do it instead in 3 to 4 months? They also have a neat application for cryopreserving animals like fish embryos so that they can just be banked in a freezer and then resuscitated anytime in the future to carry on the work. This is going to be really important for de-risking long-term engineering projects like this. It actually happened not too long ago where a group was trying to engineer fish and a control system broke near the aquarium, the water temperature changed, and all the GMO fish died. We don't want that to happen if we're several years into a 10-year genetic engineering program. And so the ability to both accelerate generation time and have this ability to de-risk projects by cryopreserving fish is going to be important moving forward. And so with that, I've reached the end of my slide deck. Thank you for your attention to this point. A lot of people to thank and acknowledge on this slide. The bolded names are the current or past lab members who have helped either with the insect EGI project or the fish EGI project. Several collaborators as well, and financial support specifically from MIT, PPC, and MAI. So the aquatic and the terrestrial invasive species research centers on the University of Minnesota campus, which are funded using funds from the Minnesota Environment and Natural Resource Trust Fund. And so with that, I will open up the floor for any questions. I believe this is going to be a moderated question session. And so the moderators are going to take control of the screen back and show that website that you can submit your questions to. And I'm happy to stay on for the remaining of the hour and answer any questions you have.
Great, thanks so much, Mike. Yes, I do have the screen showing to enter our Q&A. So we are using a tool called Slido. You can either go to the website showing, that's slido.com, and enter the event code GENES21 to participate using a web browser. If you have a phone or tablet that you want to use to interact with Slido, you can scan that QR code or again, enter the website. It'll bring you right there, and you have the opportunity there to enter your questions, and you can also upvote the questions that others have begun to ask. If you're not able to access Slido, just go ahead and put your question into the chat. And Pat is sitting here at the ready. He will get your question moved over to Slido so that it can become part of the Q&A session. I will go right up until 2 o'clock and answer whatever questions I can, or ask, I guess, whatever questions I can. Mike will, of course, be answering them. And if we are not able to get all of the questions, Mike has graciously offered to take some time later to answer the questions in a follow-up video. So I will share that link with anyone who is registered. So with that, I will get started. Right now, the leading question, Mike, was that the presentation focused on various animals, but didn't seem to mention plants. So the question was wondering if there's a limitation in this approach in regards to plants.
Good question. Yes, so we actually have strategies, we have projects ongoing in the lab to move our EGI approach into plants. but the application space is a little bit different. Let me tell you what our intended application in plants are, and I think that'll make sense to you guys, because the way I pitched it initially, that we can engineer a speciation event. We can prevent hybridization between an engineered plant that we could create and a wild version. And so our low-hanging fruit for plant applications are in GMO crop production. If you wanted to make a GMO crop and you wanted to have zero chance that that transgene can move into a wild population, you could build it in an EGI line so that none of the hybrids would survive. Now, the difficulty in using it in plants for biocontrol is you want to think about how the mating happens in plants. If we release a bunch of engineered male carp into a lake, they're going to go find all the females when the females are laying eggs, and they're going to fertilize the eggs. Though a lot of plants are self-pollinating, so they don't need that released male, sterile male. They don't need the pollen from that sterile male to propagate. And even ones that aren't self-pollinating, the plants aren't able to move around and find the corresponding female. So it's a little bit more tricky to do this particular approach in plants. And in general, I would say that most genetic biocontrol approaches will not work as well in plants, or at least they won't work as quickly in plants. That's not to say they won't work at all. And there are some groups even here on campus at the University of Minnesota exploring things like gene drives, and there's good justification for that as well. So plants are a little bit trickier just because the males aren't moving around to find out to find the females. You could think of creating an EGI plant line, finding a way to harvest a lot of its pollen, and then going around and either spraying that pollen on an area or having volunteers go on and wipe that pollen on the plants. But the timing has to be just right on that, and it's a little bit more challenging to practically implement.
Great, thank you. And I don't think I'm surprised by the next most popular question, which is wondering about this is a tool for potential eradication of zebra mussels.
Yes, so many people have approached me on this, and I would love to work on zebra mussels. But here's what I need from you guys. I was trained as a bacteriologist. My lab is more a genetic engineering lab, but we learned how to work with carp. We learned how to work with flies. A big first step is just learning how to grow and work with that organism in the lab. So for example, for zebra mussels, we would need to have a lab facility that's not just one where you can go out, collect a zebra mussel from the field and keep it alive in a tank, but you need to be able to go through many generations. And you want to be able to do that with fairly good success rates too. So zebra mussels right now, they broadcast spawn, they'll broadcast eggs and sperm into the water. You'll get the fertilization and embryo formation. My understanding though is that that's pretty easy to do in the lab. But if you had 1,000 living embryos, the fraction of those that will actually settle and start growing into a mussel, even for the best aquaculturists, is still pretty low. Until we get better at doing that, it's going to be technically very difficult for us to introduce this technology to zebra mussels. If we do though, the application space is amazing. One of the neat things about broadcast spawners like that is you could do genetic biocontrol without ever having to release the biocontrol agent. You could just put a cage of engineered zebra mussel males into the body of water in the time when they're about to spawn, and they're going to broadcast that sperm, which is the pesticide, and any egg that gets fertilized by that sperm won't develop. And then you can just pull that cage out, and you didn't have to do an actual release of the zebra mussel. So the potential applications are really exciting. I would love to meet somebody who's really good at zebrafish husbandry, because we have all the tools needed. If you give me a zebrafish embryo, the needles that we use to poke holes in carp embryos will work in zebrafish embryos, and we're happy to take some shots on goal doing that. So I think it has a lot of potential, but it's just, I need to find a good zebra mussel biologist.
All right, thank you. So the next question is wondering about biosecurity and if there's additional biosecurity measures that need to be put into place when using a tool like biocontrol to make sure that eradication stays on that local level, or at least where it's targeted.
Yes, so on a slide that I shared earlier that kind of described the parameter of persistence or invasiveness for different genetic biocontrol approaches. The labs developing technologies on the right side of that scale that are really invasive, there's a lot of rules in the field for how secure even your lab needs to be, and multiple redundant layers of security to make sure you don't accidentally release one of those to the wild. And there are a lot of people talking about how do you take a gene drive and handicap it in ways that would prevent it from being able to spread beyond the target population. However, the genetic biocontrol applications on the left side, the ones that are less persistent, like sterile release technique, those don't have as much concerns around it getting out beyond the local area, in part because they require these repeat releases. And if it ever goes somewhere you don't want, you just take your foot off the gas. You stop releasing the control agent and the population will rebound. I mentioned that through a huge effort, New World screwworm was eradicated from North America, but they're still releasing millions of sterile New World screwworms in the Panama Isthmus every year just to prevent them from crawling back up Central America. And they need to do that. And there's not a danger that those releases in Panama are by themselves going to spread down into South America. So the answer to your question is it's a great question, but it's one that depends on the specific type and the properties of that genetic biocontrol approach.
Great, thank you. The next question is, are there options for using some of the heritable controls of more long-lived animals that can happen on a useful time scale?
So it's a question. I don't know what a useful time scale is. That might be different to every person. But that's, when I talked about considering the biology of the target organism, that's a really important parameter. And I showed you data for carp and I showed you data for flies. Those are about as far apart on the time scales as you can imagine. Carp can live in the wild for 50 years. And so the application and the duration and frankly, the time it would take to decrease the wild population is going to be much longer for carp than it would be for an insect. Insect populations tend to spring up and go down seasonally. And if you can start the control early in the season, you can keep it suppressed. Actually, there's a subsidiary of Google called Verily that developed, they had a program called Bug Out, and they did a field trial in Sacramento for not a genetic control, but something that's really close to sterile release, where they use an incompatible strain of mosquito. And they showed really nicely that by starting early in the season before the mosquito population spiked, they were able to, sorry, I'll go this way for you guys. They were able to keep the population as time went very low. And the wild population in the non-treated areas spiked up as expected. In carp, do the applications differ is one of your parts of your question. And yeah, they do differ a little bit. It's known that carp, for example, there's a threshold of carp that a body of water can tolerate before the ecosystem starts degrading. I forget the exact number. It's something like 100 kilograms or 20 kilograms of carp per hectare. Forgive me if I'm wrong, Shemek, on that. But there's a known level. And if you're below that threshold, they don't really damage the ecosystem. So you could imagine, we're seeing Asian carp now coming up the Mississippi River Valley. You can imagine using a sterile male approach as a prophylactic, where you would introduce some of these sterile males that can't reproduce on their own into a body of water that doesn't have an invasive population yet, so that the first few females that swim into that body of water are more likely to mate with those males and be extinguished before they have a chance to really take hold. So for long-living organisms, you could think about applications like that. That wouldn't really make sense for short-lived organisms. But yes, there are differences depending on longevity.
All right. The next question is wondering about if there's been testing or studies looking at the acquisition of the genetically modified host DNA sequences that prevent it from moving, or from that modified DNA to move to a non-target species.
I would say, I understand the question. So the question is, will the genes jump to different organisms? And well, I don't know of specific studies looking at genetic biocontrol agents. Biologists have been looking at genes jumping between organisms for decades. And there's really not a lot of evidence that genes will jump from a carp to a walleye. that type of horizontal gene transfer doesn't occur in wild fish. And if it doesn't happen in wild fish, it wouldn't happen in the genetically engineered fish, because genes are spread by mating, and the biology of the genetically engineered fish, they're still going to mate with their like kind.
Great. All right. And then when you were talking about the EGI breeding, the question was wondering about the sex that you're releasing, and if you're releasing a male and a female, would that then produce offspring that could continue to breed with other EGI?
It would. And so there's a few, you know, whether that's a failure mode or a feature depends on how you look at it. There are some scenarios where you could imagine using males and females strategically. For example, if you floated a cage in a body of water, and in that cage or in that bay that you had netted off, you had male and female EGI, then you might be able to get away with releasing just 1 or 2 breeding pairs. And every season they would produce, let me take a step back. I forgot to mention an important point. There are ways that we can control the sexing, so what survives, male or female. And if you combine something like a sex ratio biasing approach with EGI, that's an approach that we either call SIMS for self-sorting incompatible male, or SMART for sterile male accelerated release technology, where you can have a version of EGI that in the lab you can include a special chemical in their media, in their food they eat, that would allow males and females to be born. But out in nature where that chemical isn't present, only males would be born. And the advantage to this is you could have a few breeding pairs that you have in a cage somewhere, and they can create offspring that can swim out of the cage that would only be sterile males. And so it's a way to kind of do a field amplification where you wouldn't need to release as much. Now in carp, I don't think that makes as much sense, but in some insects that makes a lot of sense. But I would say that yes, that is a potential, that if you release males and females of the same EGI line, they could reproduce if they mate with each other in nature.
All right. The next question is wondering about, I guess, like how far along the line you can pick something up and move it to another target species. So the example they have is silver bighead grass carp. So if those are close enough genetically where you might be able to have something develop that's very easily and quickly moved over to the next species, or kind of how far down the line do you have to restart as you move from species to species?
That's a great question, and it's your ability to predict how well one design is going to work in another organism is, we think, directly related to kind of the evolutionary distance between them. And that's one reason why now we think going from one species of carp to the other, that's a really close jump. The evidence that we have going from one type of insect to another is that the genes that work to overexpress for EGI, the genes that drive lethal overexpression in one insect, tend to work in other insects as well, even as diverse as from flies to mosquitoes. And so that's why we think, we're not very confident that our model system in flies, that we can just target the same genes in carp, but we think if we have an example fish, that's as close as we need. And once we get it to work in any fish, it'll be fairly easy and straightforward to port into other fish. It still takes time though. It still takes time to move the genetic components into that new species of fish. There are some invasive species which potentially faster. If you have an EGI line and it's one of these sister species that's so close that you can actually do crosses between the two groups, then we might be able to move the components over with a really kind of Rube Goldberg-esque series of controlled matings to eventually bring all the components together in the new organism. But for the most part, it requires going through that approximately 5-generation strategy.
All right, great. Thank you. So I'm just noting the time, and I see we have 1 minute left. So I'm going to stop Q&A for now. Like I said, Mike offered to wrap up and answer some of these questions offline later. So that will be available as another video recording, and I will share that out to everyone who registered with a link. So I'm going to advance my slide quick. and give my thanks. So thank you to everyone who joined us today. We really appreciate you coming and learning. Thank you, Mike, for sharing your expertise with us. Your presentation was wonderful. I think there's some great questions. I'm really actually excited to hear the answers to some of these other ones that are on deck. We do record all of our webinars, so this will be available at on our YouTube channel, which you can find at z.umn.edu/AIStube. And that takes about a week because we do a transcribe. Yeah, Mike, sorry, do you have a...
So a technical question for you. So
Yeah.
just so the community knows, my plan to jump back on and answer the questions is at 5 p.m. today. If the Slido stays open, I think we could welcome people to submit questions that they didn't have a chance to write in yet. You can do that until 5.
Oh, that's great. Thanks, Mike. Yes, yep, it will be open. So feel free again to use that at slido.com. The code is Jeans21. If you think of a question between now and 5, you still have a chance to get it answered. That's wonderful. So yep, so watch your inboxes. I said it takes about a week, and I will send out a notice through Eventbrite where you registered to see the link to view the recording and the answers to the remainder questions. I do have email addresses showing as well, both for Mike and for our program if you have questions for any of us. So with that, I'll give my final thanks to all of you, and again to Mike for having us. Have a great rest of your day. Okay, so I am back now with Mike Smansky to go through the questions that we were not able to get to during the live webinar today. So thanks again, Mike, for taking the time to come back and visit these extra questions. I'm going to start working through the list and ask them to you. The first question that I have comes from Scott Ballantine, who wanted to know if you've considered other means of inheritance as targets for engineered biocontrol, like prions, transgenerational epigenetic changes via siRNA, et cetera.
So it's a great question, and I wanted to thank Scott for the question. I also would be remiss if I didn't throw Scott some credit by name. There was a question earlier about zebrafish biocontrol, and Scott is the only brave person I've met who's actually tackling that problem head on. So it's normally out of convention to talk about other people's unpublished work when you're talking, but since Scott asked the question, I wanted to mention him in that. And so that his question is about using other approaches to create heritable changes in an organism that don't require genetic engineering. It's a great question. Scott, you're still the only person who I've heard propose that. I have, so prions are interesting. I think that's a little bit more dangerous potentially, because engineering prions to exert that kind of a change is more akin to engineering an infectious agent, and the dual use aspects of engineering infectious agents is something that I stay away from, and most people stay away from for population control, genetic control applications. But your second approach of using epigenetic inheritance, which for those of you who, if that sounded like a foreign word, epigenetic inheritance is changes to the structure of the chromosome, usually by adding different chemical groups to it that don't change the sequence of the DNA. But they can still lead to changes in behavior, form, or function of the organism. So that would be a way that you could alter an organism's biology without actually editing its genetic code. And so that is a really interesting idea. I still haven't quite figured out how it will work, but I think sometimes as a scientist, you get a sense for what a good question is to pursue. And I'm glad that you're chasing that one down, because I think it has a potential to be really impactful.
Great. Okay, the next question is in regards to spiny water flea, and wondering if this is a technique that could be used on an organism like that. The nuance being that because spiny water flea also reproduce clonally, wondering if that is a limitation in this type of technique.
So great question again. For most genetic biocontrol, they work best on sexually reproducing organisms. That's another reason why someone had asked a question earlier about plants. If plants are self-pollinating, that's like a big roadblock. So asexual reproduction would severely limit the effectiveness of genetic biocontrol. With that said, I don't know enough about the biology of spiny water flea, and if it's an organism that can reproduce both sexually and asexually, there is still some potential then for genetic biocontrol to be effective during using the asexual part of its life cycles.
Great.
Sorry, using the sexual reproduction part of its life cycles. I might have misspoke there.
Perfect. And for those who aren't familiar with spiny water flea, it is an organism that does have both sexual and asexual means of reproduction. Okay, so on to the next question. Genetic engineering is fine for honorable people, but how do you control or limit its use by people who might have like a more malevolent purpose behind it?
So it's a great question again, and thank you for asking. It's something that a term of art that we use is called dual use technologies. You know, anything that you're doing and the intention is good, if there's a potential that same thing or a very similar offshoot could be used for bad, we call it dual use technologies. And funding organizations, the federal government, academic institutions are really sensitive to dual use technology. One thing that I want to highlight in your question, and this is often misunderstood, is that in my opinion, the technologies are neither good nor bad. Applications of technologies, specific applications, can be good or bad, right? So we all use computers for good every day, but that doesn't mean you can't use a computer to do bad. Genetic engineering is a technology, it's a tool. It can be used to do good, and you could think of scenarios where it could be used to do bad. Now, the assurances I can give you are a fewfold. One, genetic engineering is still quite hard. It's hard even for the best and brightest scientists in the world to engineer complex systems and get them to work. But it is getting easier every year, so it's a worthwhile question to consider nefarious uses. What I will say is that the funding organizations that pay for research to be done have been quite forward thinking in making sure that genetic biocontrol research is done in combination with what we call LC research, which is looking at the ethical, legal, and social implications of your research. And so looking for ways where it could be co-opted for nefarious uses is an important thing that the funders want to happen in parallel with the research development. I'm of the belief that if you want to keep a technology used for good, you need to be at the forefront and pushing the boundaries so that the best people in the world at knowing how to use and detect those technologies are using it for good. And so, good question. It is something that is thought about, and it's part of responsible research is thinking proactively about those and how can we mitigate non-desirable uses.
All right. Okay, and so our next question is wondering about some other uses for genetic engineering. So do you know if we can use genetic engineering to make animals that we use for food more nutritious, and in the future perhaps would not need to use hormones or other substances in the reproductive efforts for those animals?
Yes, that is a potential application. I would say, to give a longer-winded answer, 20 years ago, biology went through a phase change with the advent of reliable and easy and fast and cheap DNA sequencing technologies. It was driven mostly by the attempt the initiative to sequence the human genome. And with all of these new abilities to read DNA easily and cheaply, every biologist is using these technologies now in their field of study. More recently, we're going through another phase change, and that's the ability to write DNA is getting cheaper and easier every year. And that doesn't mean that genetic engineers like myself are just saving money. It means that we're able to engineer more quickly and kind of turn the wheel faster. And so it's a really exciting time for biology because you get the feeling like the sky is the limit. Biology is kind of shifting from a descriptive science that looks to explain nature and understand nature and how it works to an engineering science where we can ask a lot more what if questions. What's possible with engineering biology? And so all those applications that you mentioned, engineering food organisms for increased nutritional or making them more efficient or less damaging to the environment during their husbandry, those are all potential applications. And in fact, that Pew survey, that if you look back through the video, roll it back to probably about 25 minutes in, I think the third or fourth application that was on that Pew survey was just that. using genetic engineering to increase the nutrient profile of farmed animals.
Great. Okay. So the next question asks, if you eradicate a species, another species will replace it in that vacated niche. So how does that create a risk of unintended consequences caused by the new species instead?
Yes, so I like that question. It shows a very deep level of how biology works. Nature abhors a vacuum, and if you remove a species from an environment, something else is going to take its place. But the thing I'll point out, I think the early applications of genetic biocontrol are going to be for invasive organisms. And that doesn't mean organisms that are only invasive, but for example, the mosquitoes, Aedes aegypti in the United States that spread Zika and dengue and other arboviruses, it's non-native to the regions that it lives in the United States. And so if you're using a technique like this to eradicate an invasive species, then you can argue it didn't belong in that niche in the first place. And hopefully what would fill that space, that vacuum that's left behind is, or are, the native species that it displaced in the first place. I think I used the word place too many times in one sentence, all as part of different words. But the point is, I think that that's a good thing to think about. But if we're using this technology for invasive species, we want to create that void, and we want to do what we can to foster native organisms to fill it.
Excellent. Okay, the next question reads that given there are millions of silver carp in the Illinois River, have you looked into many genetically engineered carp, or how many, sorry, genetically engineered carp would need to be released? Fish generations, keeping them in that fish generations are on the order of years versus hours for something like flies.
Yep. I think my opinion on tackling an invasive species like carp is you need to use the best-in-class physical methods, the best-in-class chemical methods, and the best-in-class biological methods to solve that problem. One of them isn't going to work by itself. We know that, and this isn't specific to your question of silver carp in rivers, but I know that common carp in lakes, they can congregate in the winter and they can be trapped or netted with fairly good efficiency, or you might reduce the numbers by 95%. Now the issue is that 5% that remains is pretty quick to reproduce and refill the lake with carp. And so I think using a combination of approaches where you wouldn't want to rely on just genetic biocontrol, because that would require, as you mentioned, the release of a lot of engineered biocontrol agents, reduce the numbers first so that you can get by with using a smaller number of released biocontrol agents.
Yeah, great. Thinking of integrated pest management, right?
IPM, yeah.
Yeah. All right. So the next question is wondering, again, this is another a question about other applications for this type of work. Is there any genetic engineering research that's being done to help organisms be more resistant to environmental changes such as climate change, or, for example, in helping organisms become resistant to harmful pathogens?
Yes, good question. There is work being done, and I can point to several specific projects that, again, maybe I'll throw out some credit where credit is due. Maciek Maselko, who is a previous postdoc in my lab, who is highlighted on my acknowledgement slide for developing the EGI approach in the first place. He's currently a professor in Australia at Macquarie University. And I know that down in Australia, they have a lot of attention to using genetic engineering approaches to protect the Great Barrier Reef and corals, for example. So that's one clear application where GE technology could be used to save reefs around the world. We have another project ongoing in my lab, which is meant to engineer soil microbiomes. So the bacteria that live in soil, and this doesn't necessarily mean genetically engineer. It could be using unique combinations of microbes that are existing in the soil already, but engineering soil microbiomes to create cropping systems that are more resilient to climate change and the diseases that spread due to climate change. And so there are many attempts to do this. And I think there's another question that's going to come up later that talks about resiliency to pests. So I'll answer that part of your question in a bit.
All right, I was gonna scroll down quick and find it quick, but it's not showing on my screen, so we might have to come back to it.
We'll get to it.
Yeah, so the next question in my list is wondering if you could make an EGI strain that was also conditional lethal to one sex to make it easier to release just the one sex.
Yes, and thank you for asking that question, because I feel like I stumbled through that part of a question before the break, where the question was about, what if you released males and females of your engineered genetic incompatibility? But the term you use, conditional lethal, is exactly what we're trying to do. I tried using other terms to describe it, but what a conditional lethal is, for example, we have insects where if you include a small molecule called tetracycline in the growth media, both sexes survive. If you remove that small molecule, then only males survive. And so that's an example of conditional lethal. And we can combine that with EGI. And in fact, another one of the forefronts is with all of these myriad of genetic biocontrol approaches that exist, when you start combining different genetic biocontrol approaches, like a sex ratio biasing and a gene drive, or a gene drive and an EGI, then the behaviors are sometimes unexpected, but in a good way, in a beneficial way, in a useful way. So yes, those things can be combined.
Great. Okay, next question is about regulatory processes, and wondering about, in your presentation you had mentioned like FDA, USDA, EPA as places where you might need permits. So they're wondering in particular what regulatory agencies do permitting for aquatic invasive species, and wondering how it is that FDA fits into that suite of regulatory agencies for that space.
So great question, and it's one that I've engaged with regulators often, and we know that it's a policy space that's quickly changing. This is a new set of technologies, and 10 years ago, 15 years ago, there wasn't really a good path forward towards regulatory approval. The companies that are leading the drive on genetic biocontrol in insects are pushing those boundaries, and there have been regulatory approvals for field trials of genetic biocontrol agents for insects. What I'll say is that it's still a little bit murky, but it's something that those agencies are working between them, and they're working with technology developers as well to figure out what are the best swim lanes for getting regulatory approval. Right now, FDA does have approval on any genetically modified animal, except mosquitoes. They gave mosquitoes to EPA. A lot of the technologies that are being pushed right now are mosquito technologies, but I imagine that more specific organisms will be pushed out of FDA's wheelhouse as we move forward. But I think some aquatic invasive species might stay in FDA. For example, carp is an edible fish. A few summers ago, my lab had a summer research party where we caught and ate a bunch of carp, and the slogan was, if you can't beat them, eat them. That's actually a biocontrol approach that's been explored in Australia. If you just make it really good recipes for carp, will that incentivize people to go out and catch them all and deplete the populations that way? So the fact that some of these invasive species are consumed by different groups of people would probably leave an organism like carp within FDA's wheelhouse. But again, I'm not part of FDA, so I can't speak on their behalf.
Sure. I think that is a good lead up to the next question. And I'll apologize, during the live, I sent a chat to get clarification on it, and I didn't catch the response before we wrapped up. But I think what the question is wondering what some of the regulatory rules are that should be passed. And they said by passed, I mean successfully completed. What I think they were asking was what types of regulations need to be put in place for this technology to be live. But if there's more to it, feel free to expand on that.
Yeah, I think the high level regulatory policies that we have work, and that's that they want to make sure that new technologies are safe to humans, safe to the environment before they approve release to the environment. And so I would say that one thing that we need domestically from an infrastructure point of view is it would be great to have more domestic infrastructure that allows for contained testing of technologies like these in field relevant environments. So Right now we have small insectaries, small growth chambers where we can do the lab scale tests. And there are some contained greenhouses. The Minnesota Department of Agriculture has a biocontained greenhouse facility on our campus. But building out infrastructure like that, which will allow field relevant testing of genetic biocontrol to generate data that will help regulators make their decisions. I don't think we need necessarily new regulatory laws passed. We just need to make it easy for the regulators to make informed decisions based on science and data.
Wonderful, thank you. Okay, the next question is one that I enjoyed because I wonder if your talk is inspiring a future genetic scientist. And the question was wondering what type of university someone might need to go to if they're hoping to work in genetic engineering.
It turns out you have to come to the University of Minnesota. No, just kidding. You can go anywhere. Scott Valentine, who I mentioned, is at University of Wisconsin River Falls. There are people working in this area all across the country, all across the world. And one thing that I'll say is that it requires diverse skills from molecular biology, genetic engineering, genetics, ecology, population genetics, regulatory policy, simulation modeling. It takes a lot of people with diverse skill sets to get one of these technologies across the finish line. And so I think that's great that you're thinking of going to university and keep doing that. But the truth is, if you have your sights set on being someone who's working in this space, you have a lot of options available. You can pick whatever university suits you best, and I'm sure you can develop the foundational knowledge that will allow you to move in this direction.
That's great. But yeah, if they want to work with you, then of course they need to come here to the University of Minnesota, right?
Yes.
All right. The next question is wondering, or it's phrased as, isn't there some evidence of genes being horizontally transferred from crops to non-crop species? For example, in Roundup resistant crops. I think this is in response to a question that was asked during the live session earlier today.
It's a great question, and it's the misunderstanding is the word horizontal gene transfer. So yes, genes have transferred from crops to non-crops. The term of art horizontal gene transfer usually is referring to transfer of genes that doesn't go by way of sexual reproduction. But the gene transfer that you're referring to in this question is actually from sexual reproduction. So it turns out that there are wild varieties that are close enough relatives to domesticated crops that you can still get cross-pollination, fertilization, and hybrids forming. And the reason that's worth mentioning is that that's exactly the application for EGI in crops that I mentioned earlier. I didn't get into the specifics, but for example, one of the crops that has not been made Roundup ready is well, it's not been deployed at least, is Roundup ready rice, or herbicide resistant rice, we'll call it by a general term. One of the reasons for that is that there are weedy varieties of rice. So one of the reasons they grow rice under water in rice paddies is a weed suppression technique. And one of the major weeds that can contaminate rice fields or rice paddies is a weedy variety that can actually cross-pollinate with the crop. So if you made an herbicide-resistant version of rice, it might be effective for the first season, but then you'll get some cross-pollination where that herbicide resistance gene goes into the weedy variety. Now all of a sudden the weeds are herbicide resistant too, and it's not a useful trait. And so if we have ways to keep transgenes in crops using barriers to sexual reproduction like EGI can afford, it can actually prevent that movement of transgenes from crops to their close relatives that are still close enough for sexual reproduction to occur.
Interesting. Okay. Can genetic engineering be used to change an organism's ability to carry a virus or pathogen? So not targeting a population control aspect, but more if an organism carries a disease or virus and then death of that individual happens.
Great question. Yes, it can. Omar Akbari at the University of California, San Diego has demonstrated with really elegant research that they could, not quite what you said, it's not going to kill the organism that has the virus, but they can prevent the organism from having the virus. So they can immunize using genetic engineering mosquitoes against dengue virus, so that mosquito can't acquire and transmit dengue virus. And I think that that's a really interesting application, and it feeds back into so many of the other really bright and focused questions you guys are asking. For example, one of the earlier questions was about what happens if you eradicate an organism locally, what's going to fill its void? Well, an alternative to that is we don't need to eradicate some organisms. We just need to change the part about them that we don't enjoy. So a mosquito that transmits disease, you don't need to eradicate that mosquito if you can just make it unable to transmit disease. And if we understand the molecular biology of disease transmission, we can use gene drives to replace a population. Instead of eradicate it, just replace it with a version of that same organism that can no longer vector disease.
That's really interesting. And it reminds me, I think Bernie in the chat had made a comment about potential applications in ticks, and I suspect this is kind of where the line that he was thinking is in disease-bearing ticks, if there's some opportunities there too. Okay, so that brings me to the final question. And I just noticed a timestamp on this. So Doug, you snuck it in just in time, 5 minutes before we started this. And so that last question is wondering which of the genetic biocontrol tools that you talked about might be best for emerald ash borer, and is that a species that's under consideration for this type of research?
That's a good question. I don't know the biology of emerald ash borer well enough to give a very educated response. I think that, so in our experience moving from zebrafish to carp, zebrafish is something we consider a model organism. It's easy to work with in the lab. People have been working on it for decades. Carp, there were already genetic manipulation protocols established for working with carp, but it was still 10 times harder to do genetic engineering in carp than it was in zebrafish. And for a lot of these insects like emerald ash borer, there's going to be there needs to be upfront investment in just learning how to do genetic engineering in that specific organism. It's a little bit of reinventing the wheel. There would be some great step change innovations possible if someone came up with an approach that just worked for every insect. But in my experience, if enough funding and attention is given to develop genetic tools in a specific organism, it's usually successful in the end. We have a lot of possible strategies in the mosquitoes or the flies that I showed you. We inject them with a glass capillary. There's some organisms where you can introduce DNA using something called electroporation, where you basically electroshock the sperm or eggs and you can introduce the DNA that way. And then sometimes we can leverage vectors as well to introduce the DNA. For example, in plant engineering, we use a bacterium that can naturally inject DNA into plants, and we just arm that bacterium with the foreign DNA and it'll introduce it to the plant for us. And so a lot of different strategies are possible. I don't know what the best would be for emerald ash borer, but I know it's a devastating invasive species, and ultimately I mentioned before, the sky's the limit for synthetic biology. The sky's the limit. There's a lot of unsolved problems in invasive species management too. There is a bit of a zero-sum game that there's a limited number of resources that's allocated to fund invasive species research. Obviously, all the invasive species researchers listening here would love money to grow on trees where they can just spend at will on any organism of choice. But I think you're right. Emerald ash borer is an important organism, and I think with enough attention and funds, there could be genetic biocontrol for that one as well.
Excellent. Thank you, Mike, and thank you again for taking the time to make it through the remainder of the questions from today's webinar. We really appreciate.
Great, my pleasure. Thank you.
Thank you.